So our next speaker is a legend and doesn't need an introduction. However, it is actually my true privilege to introduce Dr. Jennifer Lipponcott-Schwartz. Jennifer is a senior group leader at Genelia, and Jennifer and her team over the past 3 decades have really pioneered the field of cell biology and have really touched and studied all of the major cell organelles and understood their structure, their dynamics, their function, all by kind of developing and adopting bleeding edge imaging technologies. Without further delay, Jennifer, you have the floor.
Thank you. I'm going to share my screen. Can everybody see this?
Yeah, that looks good. Thank you.
Fantastic. So I want to, basically what you've heard over the last 2 talks is really spectacular technology that is really, I think, has huge potential for sort of opening up our understanding of the structure or the form of cells. But what I think is the future that we're going to have to deal with is how do we connect that form with the actual function of the organelles and the interior complexities that we're seeing inside cells. So in my talk today, I want to sort of hint at approaches for dealing with this crosstalk. How do we connect the structure, the forms that we're seeing with this technology with other types of technologies that will allow us to interrogate the function? And in doing this, I'm going to be focusing on one particular organelle, the endoplasmic reticulum, which, as you can see here, has an amazing form associated with it. The ER is the largest organelle within the cell. It forms the nuclear envelope and extends off as a tubular meshwork all the way to the periphery of the cell. Now, the ER has several fundamental functions within the cell. It's a site where all proteins that essentially are going to ultimately leave the cell through the secretory pathway or distribute among different organelles, all those proteins, which comprise about 30% of the genome, are synthesized in the endoplasmic reticulum through a co-translational pathway. Those proteins are then processed, assembled, turned over within the endoplasmic reticulum before being ferried out into the secretory pathway. Now, the issue is all of these functions of the ER, where are they occurring? We see this beautiful structure of the ER. What are the ways that we can that we're going to, moving forward, have to do in order to interrogate or relate this form and function? And so in my talk today, I want to focus on 3 different structures or forms of the ER, subdomains of the ER, and how through new technologies in terms of enhancing our spatial-temporal imaging of cells, we can get insight into the functionality of these ER subdomains. And this includes ER organelle contacts. I mentioned that the ER is the largest organelle of the cell, but it's also making continuous contacts with other organelles within the cell and communicating through lipid trafficking as well as calcium signaling to those organelles. But a second area that we don't really understand in terms of what its functions might be are ER matrices, or the 3-way junctions that interconnect all these tubes. Do they have any particular role within cells, within the endoplasmic reticulum, in terms of carrying out all of the different functions of this organelle? And then finally, ER exit sites, which are the sites where newly synthesized proteins in the ER enter the secretory pathway. Can we understand these ER exit sites to any greater extent in the context of this beautiful organelle by improved spatial temporal imaging? So let's start with the ER organelle contacts. As I mentioned, ER is all over the cell. And if one looks at the relationship of the ER with other organelles, as shown in this multispectral imaging using the lattice light sheet, so we have near 3D isotropic resolution, you can look at the distribution of 6 different organelles simultaneously and look at their co-association with each other. Now, we can do this in a more rigorous way by segmenting out the different organelles that we can see in the lattice light sheet, which is shown in that top panel here. And you can clearly see these organelles are associating with each other. And in particular, we can sort of map out an overall organelle interactome based on this type of analysis. Now, there's no question that we now have the capability of doing this using FIB-SEM at a much higher spatial resolution. But what the lattice light sheet dynamics reveals is the dynamics of these contacts, how these contacts are actually being preserved and remodeled over time. And that's illustrated in this movie here, where we can segment out the mitochondria, as shown in the left, and then we can superimpose the localization of the ER on that mitochondrial surface and look at how those contacts are dynamically remodeling over time. And it's very significant. They're changing all the time. I mean, one of the, I guess, when you look at these FIB-SEM images, as you just saw in the last, in Aubrey's spectacular talk, it's a snapshot. It's a moment of time in the cell. In order for us to understand how these dynamics, how these organelles are actually playing out over time, we need additional technologies to sort of clarify that. And that's what you're seeing here. Here are the ER mitochondrial contacts. They're abundant. They're all over the surface of the mitochondria. They're playing critical roles in allowing lipid to traffic from ER to mitochondria and vice versa, as well as calcium flux and other metabolic flux between these two different organelles. Now, if we zoom in and look at these contact sites by FIB-SEM as shown here, you can see that they are very intimate connections between the ER and the organelle that it's contacting, in this case, the mitochondria. And that's mediated by specific tethering factors. One particular tethering factor that plays a very important role in tethering ER with different organelles are members of the VAP family proteins. And VAP-B in particular can reach across and make contact with particular proteins, in this case, PTPI51, in the mitochondria. So one of the things that we were interested in trying to understand is what's the dynamism of these proteins at these contact sites that allows for the overall contact sites to show a type of dynamism across the surface of the mitochondria. And so to address that, we decided to see if we could perform single particle tracking of these contact site tethering proteins in the context of their movement of the ER relative to the contact site distribution. So 2 postdocs in the lab, Chris Obera and Johnny Nixon-Abel, have developed this single particle tracking technology. What's key for this to work is having a bright photoconvertible probe to allow the tracking of the individual molecules. In this case, what you're looking at is a calreticulin molecule, which diffuses freely within the lumen of the ER. And it's tagged with a halo tag that we can then sort of switch on with a very bright photoactivatable Janelia dye. Key for this analysis is us also introducing as a background an ER resident protein so that we can see the ER in the context of these motions. And so what you're seeing here is just free diffusion of these molecules throughout the ER. They're not undergoing any type of flow that we can see, and they're not being retained in any fashion. Now, that was really the behavior of the cow reticulum is very different from what we found for VAP-B. VAP-B, as I mentioned, is the ER mitochondrial tethering protein. And what we found for VAP-B is that although it can diffuse freely across the surface of the ER, when it comes in close contact with the ER, it undergoes significantly altered trajectories. And that can be seen in this bottom single VAP-B trajectory, where you can see the individual molecules diffusing rapidly, and then suddenly it comes into a region where it's hovering. It's not immobilized. It's undergoing very sort of tiny sort of step sizes. Now, those step sizes seem that altered trajectories is coinciding precisely with sites where VAP-B is interacting with mitochondria. And we can see that by introducing both an ER and a mitochondrial marker together with our VAP-B single particle tracking. And what you can see in these movies is that when VAP-B comes into regions where the ER is contacting the mitochondria in red, it slows down dramatically. In fact, if we create a normalized localization probability map, looking at where VAP-B is spending most of its time as it moves through the ER, that's shown in the right-hand panel, you can see that there are hotspots of localization at these ER mitochondrial contact sites. Now, this is super resolution because we're tracking, we can fit the centroid of the VAP-B, individual VAP-B molecules that we're tracking. And from that, we can get a spatial map at super resolution of all of these molecules on the surface of that mitochondria. And that's what you can see here to the right. And you can see that they're distributed all along that surface in very precise punctate sites. And what we find when we compare the size and the overall shape of these particular contact zones is that they're very much similar to, they resemble the contact sites that you can see by electron microscopy as shown in this lineup here. Now, one of the exciting things that we're doing now in this project is looking at other ER mitochondrial contact proteins to look to see whether they co-localize with VAP-B and whether they show any type of sort of spatial heterogeneity at these contact sites. This is the type of technology that we believe has the capacity to allow us to do that and to also allow us to be able to understand how these contact sites are changed under different types of conditions. For instance, when cells are starved or when cells undergo other type of stress conditions. Because we have a handle on the dynamism of this system using these technologies, we can address these types of questions. So I now want to move to what the heck the ER junctions, the three-way junctions and the matrices that are so characteristic of ER, what might they be doing in terms of ER function? Now, we really began appreciating the extent of these matrices or these three-way junctions when we had the ability to do high-speed imaging. And for that, we were very fortunate to be able to team up with Eric Betzik's group who had developed the GI-SIM system where using grazing incidence so that you're only looking at a single plane, you can image very, very fast and and combine that with structural illumination microscopy to be able to look at the ER in a very highly dynamic modality. And when you do that, what you see is that the tubes that comprise the ER in these peripheral regions is undergoing is undergoing oscillatory motion. That in turn drives changes in the position of these three-way junctions such that they can sort of collapse together into what we call ER matrices. Now, looking at the effect of inhibitors like ATP depletion or GTP depletion, we could conclude that cytoplasmic forces must be in some way mediating this process. We know that the ER can pull itself out on the microtubules, and the actin cytoskeleton can also push the ER around. But recently, what's been very exciting is that we found that vimentin filaments seem to be playing a very important role in sort of driving this sort of clustered organization of the ER that creates these ER matrices. So what are these ER matrices doing? Well, a clue really came from this vimentin work because we know in the literature that vimentin can actually interact with certain types of mRNAs. And so that led us to start thinking of whether these ER matrices could potentially play a role in mRNA dynamics. And that question has been really focused on by Hee-Jin Choi in the lab, who's decided he wanted to look at how mRNA associates with the ER and whether there are sites on the ER where the mRNA is being translated. So to do that, he created a MS2 labeled mRNA coding for transmembrane protein. In this case, saltransferase, which ultimately moves itself to the Golgi apparatus. So when Hee-Jin introduced this MS2 probe into his cells, he could see the mRNA coding for this that will ultimately translate saltransferase. The mRNA is moving, is associating with the endoplasmic reticulum, which you can see in red. Basically, there were two types of mRNA that he could see in these cells: a mobile form of mRNA and a stabilized form of mRNA. So in order to determine which of those two were actually mRNA that was engaged in translating proteins, he generated a different type of tool based on basically a SUNTAG probe where cytochrome P450, after it's in the process of being synthesized, is very quickly recognized by a fluorophore that then allows you to pinpoint sites of active mRNA translation. And so when he introduced this probe into his cells, what he found was that the mRNA that was immobile was the mRNA where the SUNTAG ER protein became visible, indicating that only the immobile mRNAs on the surface of that ER seem to be actively translating. And we could validate this conclusion using puromycin, which basically stops mRNA translation. In untreated cells, you can see by color coding, there's a large number of mRNA that are undergoing this sort of very tiny oscillatory immobile type of motion on the surface of the ER. By contrast, as soon as we hit with puromycin, the mRNA shifts to a much more dynamic modality, which we've color coded in pink here, and we've quantified it off into the right. So basically, the highly mobile mRNA on the surface of the ER is mRNA that is not translating, whereas the immobilized pool seem to be the translating pool. So is there anything interesting about where translation of the mRNA is occurring? And one of the things that Hee-Jin found was that the immobilized pool of these mRNAs seem to be primarily at three-way ER junctions or in ER matrices. So that led to the question of whether all co-translationally inserted proteins are synthesized at three-way junctions or matrices. And so I just showed you an example of a type 2 protein, but here's a type 1 protein that's coding for a type 1 transmembrane protein. And what you can see that is it's also its translating pool, which is immobile, is primarily at these three-way junctions, as is a multi-spanning protein, mRNA, an mRNA coding for a multi-spanning protein, as is a luminal protein, which is going to be secreted as a soluble marker out of these cells ultimately. So basically, all these different types of proteins that are being synthesized on the surface of the ER are doing so primarily at these three-way junctions. So that then led us to sort of head down the hallway to Aubrey's team and ask whether we could actually take a look at ribosomes on the surface of the ER. And the COSIM team kindly provided this movie where you can see that there is an enhancement of these ribosomes that are lined up in these sort of lined up patterns, which is what has previously been shown to correlate with essentially polysomes, where many ribosomes are basically lined up along a single mRNA, which is in the process of being translated. And we see an enrichment of these at these three-way junctions and at these matrices. So that then led to the question of, well, what's optimizing mRNA translation at ER matrices? Why would the ER want to translate its mRNA at these sites? And so for that, basically, we started looking back at some of our time-lapse movies, and we at the ER. And in this particular GI TurfSim image acquired by Dong Li, we immediately realized that lysosomes spend a lot of time at these three-way junctions. Lysosomes are frequently, they adhere to the surface of the ER through contact sites. You can see the lysosomes are moving along microtubules, but many of these lysosomes are spending a lot of time at three-way junctions or at these matrices. And so that led us to start wondering whether the relative distance from a lysosome might impact translation efficiency. And so for that, we looked at our type 2 protein mRNA reporter in tracking it, tracking individual mRNA, which you can see in blue, and we looked at its relationship to the distribution of lysosomes, which you can see in red. And you can see that a majority of these translating transmembrane RNA are actually proximal to lysosomes. So why might that be the case? Well, we know that lysosomes actually release amino acids. They also have mTOR associated with them, which can regulate translation. And they also can release protons, which might facilitate this process. So we look to see whether any of these, by disturbing either mTOR association or blocking amino acid release by adding a proteolytic cocktail, any of these things might perturb the extent to which these we're getting translation. And what our data suggests is that lysosomes are supporting translation through pH and proteasome, potentially proteasome activity, where they'd be releasing amino acids to allow more efficient translation at these three-way junctions. So to recap the data up to now, related to the role of these matrices, is that mRNA's coding for these co-translationally inserted proteins seem to be preferentially translating at these matrices. And we think that that's potentially because lysosomes are potentially enriched at these three-way junctions, making these regions locally rich in amino acids. And as a consequence, you get more efficient translation. So in the final part of my talk, I want to move to another subdomain of the ER, ER exit sites that are involved in protein secretion. Now, if you look at these ER exit sites using confocal microscopy, what you can see is that they're scattered all over the surface of the ER. And we're looking at a viral glycoprotein that is accumulated in these ER exit sites. The ER exit sites are characterized by COP2 coat proteins that facilitate the formation of these structures. If you look at how these cargo proteins then leave the ER exit sites and move into the Golgi apparatus, you can see that they're moving long distances. And they're moving in structures that are actually fairly big looking, that can actually send out tubules as they sort of in the direction that they're moving. So that led us to start thinking whether we could have a better way of trying to understand the nature of these structures. We know a lot about the function, but what we didn't have was a good sense of the structure of these ER exit domains and the transport intermediates that they give rise to. And so for the rest of the talk, I want to focus on some really beautiful work that Aubrey Weigel and Qilan Chang in the lab have done using cryo-SEM, FIB-SEM to reconstruct ER exit sites throughout an entire cell. And so for that, we use this cryo-SEM, FIB-SEM pipeline developed by Harold Hess, Eric Betzig, David Hoffman when he was in the lab. Cells are prepared on sapphire cover slips. They're then high pressure frozen so that they're being retained in their near-native state with no chemical fixation. And then the cells are imaged by structural illumination microscopy. These are cells that we've introduced a fluorescent probe into of the structure of interest. And in our case, we're going to be looking at components of the ER exit site. We will then free substitute to give electron microscopy contrast, resin embed and identify the cells of interest, and then start gathering block face structure, scanning EM images that through FIB-SEM, FIB trimming will allow us to move through an entire cell. And so here is, to the left, is a cell that's where we've done this for the COP2 protein, SEC23. And so all those little pink structures that you see on the left are individual ER exit sites. To the right is basically all of the FIB-SEM imaging through one of those identified ER exit sites. And so the fluorescent image that you're seeing is the COP2 SEC23 structure. Now, basically, what we could do from that is to begin reconstructing ER exit sites throughout the cell using the using the characteristic morphology of these structures. We could then start going in even into untransfected cells to map out at FIB-SEM resolution the structure of these structures. And so what we found is that these ER exit sites are comprised of a highly intertwined tubular network. That network is closely associated with microtubules. It's attached to the ER by a neck, and it has characteristic tubule diameter. Here's just another example of one of these ER exit sites where you can, that's highlighted in orange here in the context of the much more elaborate ER that's surrounding it. I want to underscore that every ER exit site that we found had this type of morphology and was connected to the ER through one or two narrow necks. They were also very much associated with microtubules. Now, one of the questions that we were interested in is what happens to these ER exit sites if we suddenly allow a bolus of secretory cargo to move into them. And to do that, basically Aubrey and Chelan basically treated cells, looked at ER exit site size, either in untransfected cells or cells where we've introduced a secretory cargo protein and released it into the secretory pathway for a short period of time. And what you can see in these two different images where the ER exit sites have been segmented out is the significant enlargement in these domains as a consequence of the delivery of cargo into them. We see both the tubule diameter becoming larger as well as the overall size of this structure. We could also map onto these structures the localization of key machinery that's involved in regulating them. Of course, COP2 is thought to be the primary gatekeeper for movement of cargo into these structures, and we could look at that in relationship to a different type of code protein, COP1, which we found was actually also at these ER exit sites, but spatially segregated from the COP2. And so based on our, in this case, it's an area scan analysis, we think that the COP2 is more localized around the base of the ER exit site, whereas the COP1 is more at the periphery. So what happens to these ER exit sites and when cargo actually starts moving out of them? And this is just 8 minutes after we've released a bolus of cargo, TNF alpha, using the rush system. You can see all these long tubules coming out of these ER exit sites. Can we see these tubules in FIB-SEM? And so Aubrey and Chelan were up to the challenge. Here are several of these transport intermediates that we know are transport intermediates because they have the TNF alpha rush structural illumination associated, imaging associated with them. So if you just zoom up on one of these little intermediates, you can see the SIM image of the cargo and see how nicely it superimposes on the actual EM structure. Now, interestingly, the structure itself has a pearled morphology. It's aligned along microtubules, and you can see it comes off the ER exit site. We can see this not only in cells where we've transfected with a cargo to allow us to verify the characteristics of the transport intermediate, but we can also see these things in cells that have not been transfected with any probe, ruling out that what we're seeing to the left somehow created as a consequence of the introduced probe. To the right, you're seeing endogenous transport intermediates that have that same characteristic pearled morphology emanating out of an ER exit site and associated with a microtubule. So in summary, what this data has really enabled us to do is get really a deeper insight into ER exit site nanoanatomy, but also into the dynamics of this system, because as I really haven't shown you, we can couple the work that I just showed you with dynamic studies, FRAP, and tracking of cargo into these structures. And what that has led us to is the following sort of model for how machinery is localized on these ER exit sites. The upper left, we think the COP2 is maintaining the neck region of this elaborate outgrowth of the ER that we call the ER exit site. COP1 is operating on the periphery of that. And when the ER exit site, when cargo starts moving out of the ER exit site, it's basically just pulling, it's protruding membrane from that site along a microtubule. So with that, I want to end and thank you for your attention. Basically, I think this field is just absolutely primed to start integrating the incredible structural insights that we're getting now using FIB-SEM and other electron microscopy approaches with dynamic advanced imaging, such as single particle tracking, multispectral lattice light sheet imaging, and the TURF, GI TURF SIM imaging that I discussed. And here are the people who did the work. I want to shout out Aubrey Chelan for the beautiful work with the FIB-SEM, as well as a thanks to Eric and Harold for their support. Single particle tracking, really spearheaded by Chris Abera and Johnny Nixon-Abel, Heejin Choi with mRNA tracking, and finally our multispectral TURF work. I want to just thank Sarah Cohen, Alex Baum, as well as Adong Lee. Thanks.
Jennifer, that was amazing. Again, on behalf of all of the participants, I want to thank you for taking the time to give us this spectacular talk. We have a few questions from the audience. I'm going to read them out. First one comes from Sam. Are all mitochondria involved in ER contacts, or is a certain percentage never involved?
Are all mitochondria involved in ER contacts? Yeah, I mean, basically so in our lattice light sheet movie, when we segmented out each individual mitochondria, we could see that there was very intimate association with ER somewhere along that surface. The more finer question, I think, is the extent to which the contact sites that you're seeing, what tethering proteins might be underlying it. And that's, I think, a question that we're really interested in now. VAP-B is certainly a component of all of the contact sites that we're seeing. But we're beginning to see that other tethering molecules may associate or not associate with these contact sites based on the particular physiological condition. Basically, there are different ways that you can sustain these contacts based on the composition of the contact site proteins.
Adila asks, do you see a difference in ribosome enhancement near the nucleus versus the periphery of the cell?
Yeah, there's definitely more ribosomes near the nucleus, no question. I mean, that's where mRNA is coming out. But what is important from this work, and I think it's really clear from the FIB-SEM images as well, is that people traditionally have talked about smooth ER and rough ER as distinct domains on the ER. What we're seeing, at least in these tissue culture cells, is that we're seeing mRNA all over the surface of the ER. But the key thing is that not all of that mRNA is in the process of translating. It's only when the mRNA is drawn into particular regions, we believe, potentially through, I don't know, maybe perhaps these intermediate filaments are playing a role. These are really open questions now. We don't know how the mRNA starts engaging in translation in these particular areas, but it could be very much many different things working together. Intermediate filaments helping to draw the ER into these matrices, lysosomes being there to facilitate amino acid abundance so that you can sort of engage the system. Our data is not supporting a direct role of mTOR, although I think we still need to look at that more carefully, since mTOR definitely can impact translation machinery.
Thank you. Gunis is asking, does the translocon complex co-localize with the ribosomes of the three-way junctions? And whether this is specific to the cell line, or if you've already, if your team has already looked at other cell types to understand this behavior better.
Yeah, I mean, we haven't looked at the translocon complex per se. I mean, we have Sec63 as a probe, but it's only one subunit of a very large structure. The translocon is a large structure. So almost certainly, I mean, the mRNAs that we are expressing are mRNAs that undergo co-translational, the proteins they code for undergo co-translational insertion. So almost certainly there'll be a translocon underneath that mRNA that we see translating.
From Etai, what are the sizes of the two particles you were imaging using the single particle tracking on the ER to visualize the mRNA transcription, or sorry, translation?
What was the size of the particle?
With the sun tags and the, yeah. Mm-hmm.
So it's just a small FP. It's a fluorescent probe, an FP. It's a fluorescent probe that's binding to the sun tag. The recognition marker on our sun tag probe. It's about half the size of an antibody, I guess. I mean, but it's fluorescently tagged, so it's going to be the size. When we track it, it's going to be the size of that fluorescent probe. I don't know whether that's answering your question.
No, I think that puts perspective to the object. And the last question comes from Thomas. What evidence indicates amino acid enrichment in your lysosomes?
When we inhibit, when we add, I'm sorry, I didn't clarify that well enough. When we added a protease inhibitor cocktail, which blocks the activity of enzymes in the lysosome that releases amino acids that allow amino acid recycling by degradation of the contents, we dramatically inhibit the translation as read out by mobility of mRNA in the region.
Thank you again, Jennifer, for joining us, and thanks to all of the speakers this morning. For the audience members, please join us for the Q&A with the speakers. Sean, Aubrey, Jennifer, well, go ahead and click on the backstage link, and there's a link to move over to the Q&A section, so that way people can directly ask questions and interact.
Great.
Thank you so much again.
Yeah.